This program is brought to you by Stanford University. Please visit us at stanford.edu.
Next speaker is Paul Norman. Paul is way out there in Perth. That's about as far away as you can get from the rest of the world, I think, in Australia. And for many years now, going on 15 years, I think he's been managing this large database. It started as a screening project for aneurysm disease and then kind of evolved into one of the world's most definitive references on the epidemiology and natural history of aortic aneurysm disease. Paul was kind enough to participate in the external review board of our SCORE project here this last January, and he was doubly kind enough to come all the way back to participate in our program today. And I kind of, along some of the themes we're developing this afternoon, is how do people get aneurysms, how do they progress, and how do we monitor them? We've asked Paul to talk about biomarkers as an indication of progression of aortic aneurysm disease. Paul?
Thanks very much, Ron, and I'd just like to thank the Stanford group for the invitation to speak today. So, as Ron said, I've come a long way. This is where I work, Fremantle Hospital, which has a magnificent view of the beautiful Indian Ocean. My office is there, and I have a view of the back of the hospital. Now, this is what it's like to get funding for aneurysm research in Australia. There's peanuts on offer. The guy chairing the review panel is not interested in aneurysms, probably doesn't know what an aneurysm is. The administrator knows you haven't got the grant and just looks on disdainfully. Talking to various people today, it doesn't sound as though it's much easier here. So, biomarkers in aneurysm progression. Biomarkers are useful in diagnosis. They're useful in, or potentially useful in, predicting progression. There's a little bit of interest in their role in monitoring treatment, particularly endoleak. And perhaps most interestingly for us today is what clues they can give us about causation and therefore what therapeutic possibilities there are. So the two things I'm going to sort of focus on are their role in predicting progression and then the possible therapeutic implications. Now, we use diameter as a marker of progression because it's the best we've got. But it's got its problems. We all know that a lot of aneurysms, particularly small aneurysms, progress slowly, which is good for the patient, but it's painful for those of us trying to study the natural history. It varies a lot within an individual from time to time and certainly between individuals. It's subject to measurement errors, so if you're using ultrasound, it's going to be at least 3 millimetres, or I should say up to 3 millimetres on repeat measures. As the rate of expansion increases, it gets near to 5 centimetres and you start losing them, so you get some follow-up loss which will bias your picture. And then, as with any aspect of aneurysmal disease, there's lots of things causing it, and those risk factors, particularly environmental ones, change. So people start smoking, then they stop, their blood pressure's in and out of good control, and so on. The sort of data you get with a cohort where you monitor it for expansion is very chaotic, so you have to be sympathetic to your statistician. Some aneurysms are stable, nothing much happens, others rocket up. A few of them have exponential trajectories, which you probably can't see in that mess there. A few of them go down a bit. A few of them are measurement error. So it's the sort of raw data is a bit of a mess. Now, biomarkers, there's a huge literature on biomarkers in cardiovascular disease, and biomarkers have their own problems. You've only to look at the CRP as an example of how controversial biomarkers are in cardiovascular disease or any process, but certainly cardiovascular disease. The two big problems are reverse causality and confounding, and CRP is a really good example of that, where there's still debate about that in terms of its value as a predictor of cardiovascular events. I work with epidemiologists, and they loathe biomarkers because they think they're simplistic and reduce a complicated disease into a molecule. Sometimes they're expensive, and as with ultrasound, they are also subject to some measurement error. So it's a really difficult thing trying to use biomarkers to study aneurysm progression, and it's not surprising because we've got a test, a biomarker, which is subject to two bad problems, reverse causality and confounding. You've got an outcome, diameter change, which is actually a surrogate for genuine progression. What the patient's interested in is what's the risk of rupture or dying, and we're using diameter change, which is an asymptomatic thing, as a surrogate. And you can't always measure that really accurately, so it's not easy. The markers for progression can be sort of split up into clinical, which Frank talked about a little bit this morning, biomechanical, which I think Charlie will be talking about, and then the biological. And I'll focus mainly on biochemical and to some extent genetic markers of expansion. This covers a little bit of what Frank said. So diameter is obviously a marker of rate of expansion. Smoking is the big one. Diabetes, a negative one, and I'll talk a bit more about that. And gender is a bit hard to work out from the literature, but may be a risk factor for more rapid expansion. I'll quickly review what's been published on biomarkers to date, and they can be divided into sort of four groups: matrix degradation products, then all the various proteolytic molecules, which we've already heard a bit about today, inflammatory markers, and then a miscellaneous group. Matrix degradation products, there's basically two have been looked at, the various elastin peptides, which Jess Lindholt has reported a few times and appear to potentially predict expansion, subsequent expansion. And then type 3 procollagen, that's actually been shown in two cohorts, which makes a refreshing change for most of the publications. So it would make sense that as your aorta starts falling apart, you can detect breakdown products, but whether they genuinely predict what's happening or just a measure of what's going on is far from resolved. Now, there's a lot has been done on the various proteolytic pathways. Again, Jess has been one of the biggest publishers here. So MMP9 and MMP2, he certainly found some evidence that the baseline levels predicted expansion. Interestingly, two subsequent studies involving Janet Powell looking at functional genetic variants that might influence lifelong levels of those two markers did not find any association with expansion. So there's a bit of a worry of failing the reverse causality test there. Other things that Jess has looked at include TIMP1, which he found no association, and then some weak evidence that alpha-1 antitrypsin and cystatin C deficiency also predict expansion. Thrombolytic pathways are important because they interact with the proteolytic pathways, and once again, the same cohort, some evidence that some of these predict expansion. This is a small cohort, so depending on the paper, there was between 90 and 120 or 130 cases followed up for 3 or 4 years, so not a huge cohort. Pi1 just reported no association, although a variant associated with Pi1 levels, again this was Janet Powell's group, found some evidence that there was association with progression. So again, no agreement between the genetics and the protein. Inflammation, obviously aneurysms are an inflammatory disease one way or another, whether it's inflammation that's causing the aneurysm or that it's just part of a wound healing process, which is an inflammatory process, and so you're measuring an effect of the disease is far from clear. Inflammatory markers are generally elevated in patients with aneurysms compared to controls, and in some cases there's an association with the actual diameter of the aneurysm, so the severity of the phenotype. But the sort of predictable markers have been disappointing. So we looked at CRP a few years ago, and there was no association between baseline CRP and subsequent expansion. Janet Powell's group looked at IL-6, no association. Jess reported macrophage inhibitory factors, some association in one paper and not in another paper, and also antibodies to chlamydia, a possibility. John Gollidge has looked at Osteoprotegrin and Osteopontin and found a weak association, and I'll come back to that in a little bit. The miscellaneous things, the sort of three main ones there, lipids. Lipids have a sort of strange relationship with, by lipids I mean dyslipidemias of various sorts, with aneurysmal disease, certainly not as important as it is for occlusive and particularly coronary artery disease. But to date, no real evidence that dyslipidemia of any sort will predict expansion. Homocysteine and glucose I'll talk a little bit about from our own data. So we have a group of aneurysms which were recruited from a screening trial, so 12,000 odd men attended, 875 aneurysms detected on ultrasound. The ones with the largest aneurysms obviously went off to surgeons to have them treated, and then we recruited 720 into a surveillance study which was followed for a median of 4 years. We got consent for DNA and in most cases sera and plasma from about 650 of the 720. And because it's a screening cohort, they're mostly small, so 79% are 30 to 39, and only a very small number were large aneurysms in the sort of surgical sense. We also have archived DNA and plasma and sera from 3,500 controls as part of a follow-up study from that 12,000. So we've got plenty of controls to compare things with. When we looked at CRP and expansion a few years ago, the CRP level was higher in the larger aneurysms, not a lot higher, but because the numbers appeared to be statistically significant, and other groups have shown that. But there was no way we could squeeze any association between CRP level and rate of expansion, which was at that stage disappointing, but probably not surprising. Since then, we've done a few other things with CRP. We've measured CRP in all the remaining controls and looked at the relationship between CRP and diameter as a continuum, bearing in mind that although aneurysms are 3 centimetres and over, from a vascular biology perspective, it's a continuum. And so this threshold of 3 centimetres is for treatment reasons, but not necessarily in terms of looking at the biology of the aorta. Anyway, sure enough, there is a sort of weak but definite relationship between CRP and diameter. So as your diameter creeps up, you have a slightly higher CRP level, which fits with having higher CRP levels in aneurysmal disease. We've looked at 3 CRP SNPs. Now, there are lots and lots of CRP SNPs of various functional significance. We chose 3 for what it's worth, and none of them were associated with rate of expansion, which would fit with baseline CRP itself not being associated with expansion. It may be that other SNPs are important, or intriguingly, there's this concept that the dilating aorta is a source of IL-6, and gets back to the notion that it's just reverse causality. So as your aorta is falling apart it's inflamed, it's producing IL-6, which then elevates your CRP, and so it fails as a biomarker. So John Golledge looked at small numbers, only 146 here, measuring osteoprotegrin and expansion, and did demonstrate an effect, although it may well have been driven by a handful of individuals over there, so not a particularly exciting effect. Same with osteopontin, very weak correlation, but we looked at a whole lot of SNPs, two for OPG and 5 for osteopontin, again with varying degrees of functional significance and no relationship whatsoever between genetic variants which might elevate the levels of those proteins and expansion. Now, homocysteines had a checkered career in cardiovascular disease over the last decade or so, and it has been of interest in aneurysmal disease. It has potential proteolytic activity, and several studies have shown that levels are higher. And we only had levels in 320 aneurysms, obviously lots and lots of controls, but the levels were higher in the subjects with aneurysms. And similarly, like the CRP measurement, there was a continuum across the whole range of aortic diameter. But when we looked at MTHFR, which is sort of the most popular functional genetic variant that can influence homocysteine levels, there was no significant association. The TT homozygotes have the highest level of homocysteine, and although their rate of expansion was slightly higher, it's only a matter of fractions of a millimetre, so that was not significant. So homocysteine's in the bin. Now, glycaemia and aneurysms, Frank was one of the first to sort of genuinely observe the negative association between diabetes and aneurysmal disease. And as he said this morning, that's been replicated in a number of studies. Janet Powell's group showed that diabetes, so the individuals who have both diabetes and an aneurysm, their rate of expansion is reduced. We've done a few things with looking further at this. So we had fasting glucose levels in nearly 3,000 men who were non-diabetic. And again, just looking at aortic diameter as a continuum, not specifically splitting it into aneurysm, non-aneurysm, there's a very weak but negative correlation. So whereas your CRP goes up with diameter, your fasting sugar goes down. So consistent with glycemia protecting against a dilating diathesis. So it's a complete mystery why this occurs. If you look at what happens to the matrix in diabetes, various things happen that increase your matrix volume. So it's a bit like the sort of thickened basement membrane that you get in glomerulosclerosis. You get glycation of the matrix, which makes it relatively insoluble. You get sort of slowing down of proteolytic pathways, which this is a sort of gross simplification of what happens. It involves some of the thrombolytic pathway changes, and then upregulation of your fibrotic sort of processes. And so that all adds up to more matrix, a stiffer, thicker wall, which, not surprisingly, may be resistant to dilatation. One thing we looked at further was advanced glycation. We've just finished this as a sort of pilot study, so fairly small numbers, about 100 controls and just under 100 aneurysm patients, and comparing those without diabetes and with diabetes and no aneurysm. The main problem is finding individuals with diabetes and an aneurysm, so we only had 27 of those. And measuring CML, which is a circulating marker of long-standing advanced glycation. Now, if you just look at the diabetic individuals, the the circulating CML was lower, so suggesting that there was less evidence of advanced glycation in those with aneurysms. Now, because we haven't got enough numbers, it's not statistically significant, but an interesting trend that may be the underlying basis for the negative association. From the small numbers we had, unfortunately, there's no evidence that that was responsible for the slower rate of expansion. It does have some therapeutic implications in that there's a lot of interest in inhibitors of glycation for diabetic patients, and it may be that those patients who happen to have an aneurysm or be at risk of an aneurysm, that risk will be increased. So that does need to be watched. Now I'll talk finally just about some of the genetic work we've been doing. I'm a bit of a genoskeptic, so this may be a somewhat jaded opinion about what genetic studies have got to offer. This is a deliberately chaotic and noisy slide. We've done more than this, but certainly these are sort of more tidier results. So this is 26 variants covering 13 genes. The number varies a bit, but hovers around 600 for most of these. And I don't know how clearly you can see the list, but there's most of the usual suspects there. So CRP, IL-6, osteopontin there, and then some metalloproteinase markers, a small number of angiotensin 1s. This is work we did in collaboration with the New Zealand group. These are slightly different. It's a smaller group, about 300, and they were slightly larger. So just perhaps ignore those. The main thing to see is each of these bars represents the three genotypes per variant. The vertical line is the average change for the whole cohort, and virtually every one of them just lines up the middle. So there's very little evidence that there's any sort of pattern in terms of certain homozygotes standing out. The ones with wide confidence intervals where they're very rare, so that's the only reason for those. The only one that did attract our eye was the Adiponectin variant there. So just to remind you, adiponectin is produced by adipose cells, and it's a good cytokine, so anti-inflammatory, anti-atherogenic, and even anti-diabetic. So it's something you want high levels of. That variant, the rare homozygote is not that common, only 7%, but it was that homozygote that seemed to have the slowest rate of expansion. It is associated with increased adiponectin levels, so high levels of your anti-inflammatory cytokine. It's associated with fewer aneurysms. This is actually the other way around, so the GG homozygote has a weak association with aneurysms, and it appears to be associated with slower expansion. Now, because of we were interested in looking further at the role of diabetes, this is a sort of second, somewhat chaotic slide, but was the only way to try and communicate it. This splits up the cohort into those who are non-diabetic along there, those who are diabetic along there, and it's some modelling splitting them up by diameter, so 30, 35, 40, and obviously as your diameter goes up, the vertical axis is the annual growth in millimetres. And so as you get bigger, your expansion goes up, same with the diabetics. If you're diabetic, this line is lower than if you're non-diabetic. And then finally there's the three genotypes. And the striking thing was that the TT effect was mostly seen in the diabetics. So irrespective of the size, the TT homozygotes had really slow rates of expansion. You see it a little bit there, but statistically quite weak compared to down here. Now, these are very small numbers once you start getting into these subgroups, so I'll just sort of put a caveat on that. It's just an observation, small numbers, and not adjusted for multiple testing. If you correct for multiple testing, your p-value just, again, goes out the window. So it's an observation that's interesting and needs replicating. The other thing that doesn't quite work is John Gollidge looked at adiponectin levels in a case control study using some of our samples, and the aneurysm patient subjects had slightly higher adiponectin levels, so it doesn't fit. So another biomarker that doesn't quite make sense. Story of my life. Adiponectin is interesting though, because there are some therapeutic implications. You can put your adiponectin level up by exercise and weight loss. So doing the right thing, part of the doing the right thing ends up being involving adiponectin. And you can put it up with various drugs. So triglyceridone is one, and then this one which I hadn't heard of but read about recently, so it's a cannabinoid 1 receptor blocker, which apparently puts your adiponectin levels up. So it is potentially something that can be manipulated pharmacologically and, more importantly, by exercise and weight loss. So I think it's something that merits further attention. So where does this leave us? We've got 20 or more putative markers or non-markers. Only studied in a small number of cohorts, so there's been multiple testing within those cohorts, and we're guilty of that. Big problem is there's been negligible, virtually no replication, and so no one knows where we stand. Part of that relates to publication bias, so the first positive gets published, someone else repeats it, it's negative. Either they can't be bothered submitting it or they know they won't get it published. And so the saga perpetuates. And there's, as I've alluded to with some of the things we've done, there's often poor concordance or agreement between a biochemical observation and a protein association and the sort of genetic markers, or vice versa. So you can't sort of close the loop in terms of making sense and getting a causal pathway. So this is Humpty Dumpty, in case you don't recognise him. And the reason he fell off the wall was he ruptured his aneurysm. And the reason they couldn't put him back together again because there was no neck. But I don't think a blood test would have helped him. And I think that's sort of where we're at the moment with biomarkers for aneurysmal disease. It's still interesting, but we haven't really made any progress. Some strategies for the future, and some of these have already started to happen. Multi-centre collaborations, and in particular some sort of coordinated replication. So replication in genetic studies is huge now. You can't get anything published without some element of replication, but we really need it for the sort of biomarker stuff in aneurysmal disease. Screening is being introduced here and in the UK, and that's a great opportunity to get large numbers of patients with aneurysms and study them and collect, where possible, collect biomarkers. And then genome-wide is partially funded in various locations, and to a lesser extent proteomics is being expanded. So they're some of the things that will help the situation. Just thank some of the many people involved in this. I won't go through them individually, but there's a lot more than this who did all of that work and statistical work. John Gollidge I mentioned a few times. Thank you very much.
Don't go away. So in his usual understated way, Paul pretty much covered the waterfront there with biomarkers. Yes, there's going to be a lot of questions. We're going to have to limit them to just a few. So, Richard?
All right, just A couple of brief comments. First of all, to the extent that AAA disease is genetic at all, or gene by environment, it's not Mendelian, and so it's very important to get away from thinking that the marginal impact of one change or one SNP or one protein is going to be very, very telling. I mean, this goes for cardiovascular disease and other things as well. Combining a variety of things is a challenge, but not an unmanageable one for statistics. If you look at your CRP by aortic diameter graph, you'll see that it looks somewhat hyperbolic. If you took logs of both of those things, you would find perhaps a linear relationship that was more firm than what you found. And just in the interest of time, I'll just make one more comment. One needs to be precise in these studies as to what's being predicted from what. What is the precise phenotype being predicted? Because it wasn't really made clear in what you said. Maybe it's maximal diameter of something, maybe not. But in any case, progress in looking for complex gene by environment interactions depends crucially on defining what the phenotype is, and that's at least as much a burden as is anything else.
So now you have some insight into the circular discussions that came up during the external review board this last January. Is ultrasound the right endpoint? Are we looking at the right way of analyzing the outcome? It's a very challenging area. So this, Paul just gave you a really excellent insight into some of the issues involved with designing these studies and analyzing them. And I should also point out that we're actually trying to work out some kind of replication cohort arrangement with Dr. Norman in the Western Australian study, as well as perhaps a few other groups, and this is an area we think that provides us a lot of opportunity to answer some of these questions in the future. Other questions for Dr. Norman? Yes.
Thank you. Very interesting talk. I'm from Dr. Fayyad's group in Mount Sinai, New York, and we're working on imaging. And well, AAA is pretty interesting, and as far as molecular imaging is involved, it seems like we're still looking for the right molecule to predict the progression. Even though I guess we're not gonna end up with one single molecule, but you mentioned elastin degradation product. Do you think that might be interesting to study in this direction?
Well, firstly, there's no way it's going to be one molecule.
Right.
It's going to be a cocktail of a whole lot of things, if at all. The matrix degradation products of various types, I mean, if you just think about it, they're so much part of the result of the pathology that I suspect you're going to have reverse causality. It's going to be very hard to predict that, to sort that out. So I'd be very skeptical that they're going to be... you want to find out what's initiating and causing the early start of the process, not when it's already underway and you've got those products in your circulation.
I agree.
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.